Welcome to session 3 video for cell and tissue engineering. So here you see in my hand some agarose powder because we're going to basically make gels. We want to take our cells from our previous labs and suspend them in a three-dimensional gel construct. So we're going from cell culture to engineered tissue culture starting from here. So we need to first weigh out the appropriate weight of agarose powder. We need roughly about half a gram of agarose powder. And that agarose powder will then have to be dissolved within an appropriate solution. The solution that we're going to dissolve that half a gram of agarose powder in is right here. It's phosphate buffer solution. We will actually need to warm this phosphate buffer solution as well, but here you just have the phosphate buffer solution, okay? And we're gonna then warm it up at 60.5 degrees centigrade in a water bath. And the reason for warming it up is obviously to enhance the dissolution and ensure that the agarose powder completely dissolves within it, okay? So once we have warmed up the phosphate buffer solution, or PBS, here we have already weighed out our half a gram of agarose powder, and now we are ready to go ahead and mix the agarose powder, half a gram of it, in 50 ml of the PBS liquid. So let's go ahead and do that. Okay, we're going to mix it. Again, keep in mind that the water has to be warmed up to ensure that the agarose powder is completely dissolved. So you can see me adding the agarose powder into the PBS. Okay, and here I'm essentially using a stirrer here, or a pipette, to enhance the dissolving of the powder, right? Essentially just mixing it. Okay. So mixing the agarose powder within the PBS, and so now the agarose powder within the phosphate buffer solution becomes my gel solution. Again, very non-viscous. I continue to put it in the water bath to ensure that the powder completely is dissolved for a couple more minutes. Now I've transferred everything into the laminar flow hood, and the agarose powder has completely dissolved in the phosphate buffer solution, or PBS. And what I'm going to do here is I'm going to essentially use this gel solution to make molds within a mold construct. The mold construct could be Eppendorf tube, or you could even use the cap. Okay, but in this case, we're going to just use well plates. But you could use the cap of the Eppendorf tube over there, as I'm pointing to. You could use the tube itself. So it's your choice of what mold you want to use. But like I said, for this lab, we're going to use regular well plates. So I'm going to go ahead and take about 2 ml of gel solution and place it in each of these wells. Keep in mind that we need to repeat this procedure for each of our experimental groups. If you go back to the protocols, those groups involve a variation in FBS usage, either 0, 10, or 30% FBS. The steps are the same, other than the fact that the media you'll be adding at the end would either contain 0, 10, or 30% FBS. So here I am adding 1 ml of gel solution into a well plate, and we need 1 more ml. So I go ahead and add that over there. Okay. So it's important to keep in mind that this is now at room temperature, and as we wait, the gel is becoming more and more viscous. So we typically want to wait about 4 to 5 minutes before we add the cells. If we wait longer than that, it will be too viscous to add and mix the cells in. So now it's been about 4 to 5 minutes, okay, about 4 minutes or so right now. So we're going to take this cell suspension, which you see here, and we're going to pipette some out, okay, Specifically, we're going to use about 200 microliter of cell suspension and add it to that 2 ml gel solution. Okay, and we add it to each of our wells where we have gel solution in it. Keep in mind that over here we still need to mix the cells uniformly, because uniform cell seeding within our scaffold, in this case the scaffold being the agarose gel, needs to be as uniform as possible. So we have to mix it in, but as we mix it in with the pipetting steps up and down, we want to make sure that we try to minimize the creation of air bubbles, because we don't want air bubbles to be trapped in there. That could compromise the viability of the cells. And you can see in this example, there are a few air bubbles there, so that's what air bubbles look like. But you do also see the pinkish media has mixed in with the gel. So you could probably try to poke some of those air bubbles out, or just be more careful with the pipetting technique. It is somewhat of a challenge nonetheless. All right, so now we're going to give an opportunity for the gels to gelate. So here we are with, after about 10 minutes or so, the cells uniformly seated within the gel construct. You can see I'm flipping the well plate over and nothing's coming out. The gel solution has now become sort of a jello-like construct. It's stuck to the bottom of the well plate. It has completely become more viscous and become like I said, like a jello. It is now a gel, no longer a gel solution. Okay, and the cells are of course suspended within that gel. So now we have a 3D instead of a 2D construct. And at this point, we can provide nutrition to these gel construct, just like we did in flask culture, by adding the appropriate amount of media to each of these wells that contain the now cellular gel constructs. Remember, again, these are mesenchymal stem cells, human mesenchymal stem cells that we've seeded within these gels. So we need roughly about 2 ml of media, and I'm adjusting the pipette so that we can put about 2 ml of media per well plate. So there we go. And again, about 10 minutes or so for the gel to completely gelate. And now that it has gelated, we can add the media to provide nutrition to the cells within the gel. So I'm adding the media, as you can see here, right? And also important to keep in mind, and you may be wondering, how do we get the cells to begin with? I ask you to refer back to lab session 2, where we used trypsinization to remove stem cells from a flask. We centrifuged it. We replaced the supernatant with fresh media to resuspend the cells. So you want to pay attention to the Trypan Blue session 2 video, where we already went through all those steps. I didn't show you how we actually recovered the cells from a flask over here, because that's already covered in video session number 2. At this point, we have provided the media for the gels and the cells that are in the gels, and we're ready to store them for another week of culture, this time in 3D gel culture for that 1 week. And that would be under standard incubator conditions at 37 degrees C and 5% CO2 in a humidified environment. Here are our valuable cellular gel constructs. Again, mesenchymal stem cells within the gel, agarose gel, and we're going to place them for a week of culture in a standard incubator condition. Okay. This concludes session number 3. Thank you.